I'm Chris Chamberlain. I work for UCB. It's a real pleasure to be here today to introduce a topic of extreme importance to all of us. The goal of personalized medicine is, of course, to make a difference for patients. That's where the value will ultimately be felt. And today, in a meeting that is sponsored by the UK Stratified Medicine and Pharmacogenetics Network and NHS England, we're here to talk about just that. Making a change at the level of practice, making a change in the care pathways that support patients in achieving the best outcomes. We're going to think about stratified medicine, what it can achieve, and how that can actually be translated through to making a difference in our everyday practice. This is an interactive forum. There'll be plenty of opportunities to have something to say, and I would encourage you to be vocal, to bring what you have to say into our discussion. So at the end of it, we'll be able to capture it and synthesize a publication that represents where we think we're going in this space and hopefully make a difference to the strategic planning for personalized medicine within the NHS. I'm told that we're recording today. If you've expressed a desire not to be recorded, obviously that's been recognized. If you are speaking, enjoy the recording box. We're not expecting...
That's for the audience for questions afterwards.
Yes, that's what I thought. Yeah, we have something far more sophisticated for our speakers, which is not 6 inches wide and bright green. We're not expecting any fire alarms, so if you hear anything, follow me in running out of the door and getting out of the building very quickly. At this stage, then, it remains just to begin the proceedings. I'm delighted to invite Munir, who you all know and who is responsible for this meeting, to tell us about drug safety and the impact on care pathways for personalised medicine. Thank you, Munir.
Thanks very much, Chris. Can you hear me? Yeah. So let me thank all of you for coming to this meeting. It is very important, as Chris has said. We were going to start off with Fiona's talk, and we thank NHS England for holding this workshop together with us. But unfortunately, we had to change the order because there's a little thing called the BRC competition going on at the moment in England, and I have to go to that workshop for 2 hours, and I'll come back in the afternoon. So I'm going to talk to you about drug safety and impact of care pathways. And I'm going to give you one specific example and take you through why we've tried to develop it. In terms of the science, you need the evidence to start implementing and changing care pathways, but also some of the issues that are arising from us trying to change care pathways. And it would be good to get your perspective on it and your experience. So why do we look at drug safety? Well, we did the biggest study still in the world, and it was published in 2004 in the British Medical Journal. And we followed prospectively about 19,000 patients being admitted to our hospitals. And what we found was 6.5% of all individuals admitted to the two hospitals were due to adverse drug reactions. And when we extrapolated that to the bed base in England and Wales at that time, we worked out that 7 800-bed hospitals could be occupied by patients with adverse drug reactions. That's 5,600 beds. So that's an enormous burden at a cost to the NHS in excess of about £750 million. So that is enormous, and 70% of those were potentially preventable through different methodologies. And if you look at the drugs which were causing the problems, these were drugs which are most commonly used. Aspirin, hugely important in terms of preventing coronary artery disease and so on, but nevertheless causes GI bleeding and other adverse effects. This was #1 cause. Diuretics, used in heart failure, various different things, but cause renal impairment, and again, important area. And the third one was warfarin, which is very widely used, and this is what I'm going to talk about, warfarin. I don't really have time to talk about what we're trying to do with this in terms of preventing that at the moment, because that's further behind, but the warfarin is furthest ahead in terms of us trying to implement changes within the NHS. So why warfarin? Why is it so important? Well, warfarin is used by 1% of the UK population, and it's likely to increase in terms of oral anticoagulation. I'll come back to the new oral anticoagulants as well, which are available. The problem with warfarin is that there's a huge variation in dose requirement. So some people may require 0.5 milligram a day, some require 20 milligrams a day. So there's 40-fold variation, and we have a biomarker called INR, International Normalized Ratio, which we monitor, and we try to keep that between 2 and 3. So if you go above 3, there's a risk of bleeding. If you go below 2, there's a risk of thrombosis occurring. And if you have an INR which goes above 3, that increases the risk of bleeding, such as in this 82-year-old lady who had a calf hematoma after 6 weeks on warfarin for atrial fibrillation. And as in our study, there have been many different studies since then throughout the world, and where warfarin is used, warfarin is always in the top 3 of drugs which cause adverse drug reactions. So there's something we need to do to be able to improve the way we use warfarin, as well as oral anticoagulation in general. And I'm going to give you some science, first of all, and then tell you how we went about trying to overcome some of the issues. So warfarin is a drug which is handled in the liver. It's broken down in the liver. It's broken down into breakdown products of metabolites, which can then be excreted from the body. And in order to break it down, the liver has certain enzymes. One of those is called cytochrome P452C9, CYP2C9. And that varies in this audience. So there'll be people in here who had very little levels of cytochrome P452C9, while others have very higher levels. And you would have thought that if you have low levels, you're not going to be able to break down warfarin, so you'll need lower doses. Now, warfarin acts on the vitamin K cycle, and there's an enzyme called vitamin K epoxide reductase complex. So warfarin inhibits that enzyme and therefore prevents the formation of clotting factors, activated clotting factors, require vitamin K for the production. And again, this varies. So people in this room will have different levels of vitamin K epoxide reductase complex. So again, if you have a low level, it's likely, biologically plausible, that you require a lower level or lower dose of warfarin every day. And in fact, that's exactly what we found. So if you look at factors that affect warfarin doses every day, well, there are many clinical factors, such as age. So as you get older, you require a lower dose of warfarin every day. And that's because our liver gets smaller as we get older. So that's something forward to look forward. We can look forward to that, can't we? Body mass index is also important. So the heavier you are, the sort of greater the dose of warfarin, and gender and body mass index sometimes go together. And then there are many drugs which can affect the way we utilize warfarin as well. But if you look at all of these things, they only account for about 10% of the variation. The majority of variation is caused by variation in vitamin K epoxide reductase complex, as I showed you, and cytochrome P452C9, which is the one that metabolizes warfarin. And so that's 40% of the variation is caused by that. So what we wanted to do was to see, well, how can we actually start bringing genetics and the clinical factors to be able to improve the way we dose warfarin so we can make it safer. And so we undertook big studies looking at the whole genome. And the two things that come up again, reassuringly, is there's just the 2 genes. So that's what we need to look at, cytochrome P452C9 and VCOC1. And again, you can see on this genome-wide study, these 2 genes are by far the most important. So that gives us reassurance that that is what we need to do. Now, if you're going to start changing doses, you need to develop algorithms. And in personalized medicine, you'll have heard about people trying to develop algorithms for all sorts of things. And so in order to develop the algorithm for dosing, you have to have a large data set. So what we got involved in was a very large international collaboration with about 20,000 patients who were captured within this collaboration, so we could actually start developing algorithms. And this was called the International Warfarin Pharmacogenetics Consortium. And we published the algorithm for the first time, really, in particular area of personalized medicine in the New England Journal of Medicine in 2009. So what we wanted to then do was to see, well, how can we actually start developing the evidence base for why this algorithm would work. So this is the warfarin dosing pathway at the moment. So if you have AF, atrial fibrillation, or a deep venous thrombosis, your doctor will decide to use warfarin. And initially, they might give you a loading dose, usually 3 doses, but the practice varies, and where you're given that varies. And I'll show you some data on that. Day 4, you come in and you have your INR checked, which is the international normalized ratio, the biomarker, and then you have your dosing changed again. And different clinics will use either manual dosing or have a computer software that allows you to be able to decide on dosing. And there's several computer systems available, RAID, Dawn, etc. And then you find when you get your maintenance dose, you will come in at different time points and have your dose adjusted according to INR. And again, different computer systems will be used. So there's already differences in the clinical pathways which are used to be able to assess how you will use warfarin. Now, if we did a survey of 30 UK clinics, and you can see that because of the complicated warfarin pathway, the dosing is actually different in different areas and different clinics. So this particular dose, which is 10 milligrams on day 1, was most common, and 10 milligrams on day 2 and 10 milligrams on day 3. But there were some people who were giving doses of 1 milligram day 1, 1 milligram day 2, 1 milligram day 3. So there's huge variation in doses which people are using in warfarin in the UK in different warfarin clinics. So that already complicates the situation in terms of there's no standardized dosing protocol at the moment. So here, if we go back to our warfarin dosing pathway, and if we wanted to introduce genetics, we need to incorporate algorithms into that pathway. So first of all, what we did was that we need to take into account that we need to give loading doses. So we used the original dosing algorithm that we developed with the international consortium, modified it to take into account loading doses. We then developed a second algorithm, which was we used INR here on day 4 to incorporate that into a dose revision algorithm. So there's 2 algorithms there, and then what you have is your usual dosing software within the NHS that you can utilize. So now how do you actually go about developing the evidence that these 2 different algorithms work and can be made to work in an NHS setting? So you have to take undertake a randomized control trial. So that's what we did. We got some funding from the EU Commission. We worked in Liverpool and Newcastle for the UK centres, and also a Swedish centres as well. And we randomised 454 patients. If you randomised into genotyped arm, you had 1 ml of blood taken and worked with LGC, which is a company in Teddington here. And through that 1 ml of blood, it would give us the results on the 2 genes within 2 hours. And those 2 genes could then be incorporated into our computer software, the algorithms, and that would tell us what the dose would be. So it could be 7, 5, 3 individualized to some patients. And we compared that to standard care, and the standard care was 10 milligrams of day 1, 5 milligrams day 2, 5 milligrams day 3. So you can see how complicated it is already. And so we did this trial. We recruited 454 patients. And then we got the results and we published the paper in the New England Journal of Medicine. And what we were looking at was a primary outcome measure, which was the quality of anticoagulation control. How close you were within the INR of 2 to 3 for the first 3 months of therapy. And if we look at the control arm, first of all, we got a very good control in the control arm, 60%. Now, at 3 months, you don't normally get that in NHS clinics. It may get to 60%, etc., over a year or 2 years, but over 60% over 3 months is actually very, very good. But that was because we were monitoring them very carefully, even in the control arm. Yet, in the genotyped arm, we actually got a better result, 7% difference, and that was highly significant. And if I just show you some data on Kaplan-Meier plots, what you see is that if you look at INR, in the control group, which was the conventional dosing that goes on in the NHS at the moment, you get an overshoot of INR, and as I said, the higher the INR, the more likely you are to bleed. Yet if you compare that to the genotyped group, you don't get the overshoot, and you get to the 2.5 kind of level, which is where you want to be, much more quickly. And again, this is seen in terms of the quality of anticoagulation control, which is time and therapeutic range. So the genotyped arm is getting there quicker, and you get a separation in the 2 curves on about day 10. So the genetics is working as early as day 10 in terms of separating the 2 populations. Now, the other thing that you need to show, we showed it was effective in terms of better anticoagulation control, but obviously the NHS, we have to think about resources and make sure it is cost effective. So we also undertook a cost effectiveness analysis as part of that. And what we see here, which is, I'm just concentrating on the UK curve, and if we consider that our threshold in the UK, according to NICE, is between 20 to 30,000 per quality adjusted life year, our cost was 6,702 per quality adjusted life year. So it's highly cost effective. And you can see that it's in terms of the cost effectiveness curves, that it reaches plateau very quickly. So that's the evidence behind it. So we felt that it was important, and we should think about how we can start implementing that. It's appropriate for commissioning because it's a public health issue, 1% of the UK. There are other direct oral anticoagulants available, and I'll come back to that in a minute. We've shown it was effective in a randomized control trial. Obviously, we hadn't got clinical outcome measures such as bleeding, but we need a randomized control trial of 20,000 to be able to do that. We've shown it was cost effective, and it will be ethical because it will replace haphazard dosing. But whenever you're looking at personalized medicine aspects, one other thing you have to consider is whether you're going to exacerbate health inequalities. And that's crucial. So this particular algorithm really only works in Northern European populations. It does not work in Indian populations, in Afro-Caribbean populations, or in Chinese populations, because we haven't had enough data from those populations to develop population-specific algorithms. So that is a limitation of that particular algorithm. So, and this will happen with all sorts of personalized medicine, and we need to take that into account. So that is an issue perhaps we can discuss later. The other thing that we obviously need to consider when we're using warfarin is that what about the new anticoagulants which are there, which are great innovation, but is there issues associated with those? Well, many big trials undertaken by drug companies that manufacture these three oral anticoagulants, which has shown that they have comparable efficacy to warfarin in most cases, sometimes superior, sometimes non-inferior. And certainly if you do a meta-analysis, there's less intracranial hemorrhage with these new anticoagulants, but possibly higher gastrointestinal bleeding because of local effects that they have on the gastrointestinal tract. They're marketed on the fact that you don't need any monitoring. You give the patient a fixed dose and they go away and they come back, but there's some issues with that as well. There are fewer interactions. There are some interactions, but fewer than warfarin. And NICE has certainly looked at the cost effectiveness, says it is cost effective to utilize these drugs. However, let's see what's happening to the cost and so on within the NHS and NHS England figures. So these are available in the public domain, so that's what I'm using. So 2014-15, the total cost of NHS England was 89.3 million for these drugs, just on anticoagulants. So that's a huge cost, nearly 100 million. Okay, the prescription spend on these new anticoagulants, diaryl or anticoagulants, is increasing. Between 2013 to 2014, it had increased by 504% for apixaban, 48% for dabigatran, and 183% for rivaroxaban. The patent expires in 2023, and the spending on these DOACs is likely to continue to increase. However, warfarin is still used, and warfarin, there were 11.5 million prescriptions last year at a cost of £20 million or £21 million, and for DOACs, there were 1.3 million prescriptions at a cost of £89 million, as I said. So there is a huge cost outlay, even though it is cost effective. And we need to start thinking, well, what is the way we can actually make sure patients get the drugs they want, such as DOACs, but also maybe use warfarin in certain populations where it may be effective without any of the problems associated with it. So again, if you consider the direct oral anticoagulants, they are cost effective, but the cost effectiveness depends on anticoagulation control with warfarin. The better your anticoagulation control with warfarin, the less cost effective they become. So that's an important thing to consider. The cost effectiveness has not taken into account the cost of using antidotes. There were no antidotes, but new antidotes are coming through. So this antidote, which I can't sort of say, and I'm not going to try to say, which is a dabigatran antagonist, will be £2,400 per dose. So if you take into account the bleeding and the cost of it and the cost of antidotes, even though bleeding is relatively rare, then we don't know what the cost effectiveness is of the combination. Although marketed as not requiring monitoring, significant number of patients have anti-10A levels done at the moment. And obviously all these drugs also require electrolyte monitoring and liver function test monitoring. And they're not suitable for some patients where there are adherence issues, because sometimes some of them have to be taken twice a day and they have a short half-life. And there's those people with abnormal renal function, they're probably not suitable for as well. And warfarin continues to be still widely used, and in children as well, it is also widely used. Now, we know that from other data published by other people that there are certain individuals who are particularly sensitive to the warfarin bleeds and so on. And this accords with the data that we produce with the genetics as well. So if you take those individuals who are highly sensitive, you can see that they have a higher risk of bleeding, in addition to what is currently used in the NHS to assess the risk of bleeding. And what this data from, which was published in the Lancet from a very big trial, suggests is that we may be able to stratify patients. So you could, if you knew what the genotype was, you could actually give those individuals with the highly sensitive and sensitive variants DOACs, while those individuals who have the wild type for cytochrome P452C9 and VCOC1, which is the majority of you, probably about 62% of you in this room, you could be given warfarin. Now, I know that in those patients, and we've got data, etc., is that irrespective of the way we dose people with warfarin, those people are probably okay with warfarin, actually, and very little happens in terms of the variability in INR. And so can we actually identify those populations and implement that into clinical practice? So in terms of implementation, the choices that we have available now, because of innovation for the new anticoagulants plus innovation in genetics, there are many choices available, and we need to make those critical choices right now in terms of implementing to the NHS. So we can switch over to DOACs. That's one possibility, or we could use warfarin only. Alternatively, we could improve warfarin dosing through the use of genetics, plus taking into account other factors, or we could stratify the choice of anticoagulants, which could be some patients get DOACs, some patients get warfarin, based on genetics plus other clinical factors we could use. And that is truly a choice for personalized medicine. And my feeling is that we can't really switch over to DOACs, everybody, for reasons I've already mentioned. We can't just use warfarin, because some patients do need DOACs. So what we need to think about is using both of those, improve warfarin dosing, but also stratify the choice of anticoagulants. So those people who require DOACs get the DOACs first time, and those who require warfarin can have personalized or precision dosing. Everybody talks about precision medicine, but precision medicine should be built on precision dosing. So this is what we're trying to do at the moment in the Northwest Coast and doing our implementation. I'm going to tell you the kind of issues that we've had with implementing within the Northwest Coast. So when you want to implement something, people will say, isn't this a research study? They say, well, no, actually it's implementation. But, well, we better ask the ethics committee chair. So we went to the ethics committee, who confirmed it was service evaluation. So that took about 4 weeks. So then we went to the letter to the medical director. So he had to approve it. Then he said, well, you better get a clinical lead to approve it. So we got the clinical lead to approve it. Then they said, well, there's no department that deals with this kind of innovation, so you'll have to go to R&D department. So the R&D departments have now changed their names from R&D to RD&I, Research and Development and Innovation departments. So you have to do the innovation. So, there's no gold standard guidance at the moment to follow, so they have to develop their own guidance and standards and so on. The problem is that if you do this in one trust, you have to do it in a different way in a different trust. And we've got approval in one trust, but we still haven't got approval in other trusts, despite 6 months of doing this. So that's number one problem in terms of implementing. So that's the first bit. So, and just keep on looking at numbers. You'll see how many I get to. So then there are the information governance processes. So you have to have a patient information, which is very important. So that has to be overseen by a lay readers group or a patient information forum, and then by a lead anticoagulant nurse, make sure they agree with it, and so on. So then you have to print those out, so you have to then go to reprographics, because they are a trust issue, and the meetings are held monthly at the Royal Liverpool Hospital. Reprographics then have to say whether they're going to print it for us or not. That's another one. Okay, now, So the other thing is, once we've got these documents that we've actually incorporated into one hospital, we're unsure if the documents will be accepted by the other trust that we're working with, and they may want their own documents and go through the whole procedure again. So we're just trying to do that at the moment. Then you want to collect data. Obviously, if you're doing implementation, you want to make sure data is collected and so on. And the data collection forms, you have to go to the Data Protection Office, the information governance leads, etc., to make sure that you are adhering to data protection standards within the NHS. And that's important, but it could be streamlined. We've had detailed discussions with information governance about data collection, and because we need to do some analysis to see that the implementation is working, we need to de-identify all the data so they can be taken out of the NHS trusts, and obviously we had to go through processes to be able to do that. And when you have staff in the information governance office with change, we have to go through the process again, explain it to them, what we're trying to do, etc. Number 3. Then you have your DNA equipment. So obviously you need to make sure the analytical equipment you have is appropriate, is rigorously tested and so on. So we undertook a rigorous testing at the university for quality assurance purposes. It took longer than initially anticipated. And the reason for that is that LGC has developed a new platform which actually does it in 45 minutes rather than 2 hours, based on forensic work that they do, and police can use it in the field for forensic examination. But when you're doing forensics, you can get an ambiguous result. That doesn't really matter to the police, because there's an ambiguous result. You'll have to have a proper DNA test for assessment. But if you're in a clinical setting, you can't really accept ambiguous tests, because if you keep on getting lots of ambiguous tests in clinical settings, and then that causes problems. So we had to work with LGC to change their assay, and so that we got rid of the ambiguity, so that we would get the result which is appropriate within clinical setting. And that did take a while. Once you've got that done, need to have the training. So LGC came across to be able to train all the nurses and so on. Because clinics are busy and they don't have specific time for training, you have to split it over several days and so on, or 2 to 3 sessions to do the training. The para-DNA has to be password protected, so nobody can get access to it, so it requires secure storage. So we had to find somewhere in the clinic to be able to store it. Because you're putting it into the NHS, you have to have, obviously, PAT testing, etc., indemnity insurance documentation, declaration of decontamination status, etc. So we had to go through all those different processes as well. And I think we're up to about 20 forms so far, but there's more coming. And then we had to do a standard operating procedure for cleaning the apparatus as well for the NHS. And that's important, I understand, for infection reasons and so on. So again, that was done as well. Then we've got the samples. We're going to do we are doing the analysis in clinics, but we obviously want to make sure that what we're doing is appropriate and is quality control. So we are taking the samples back to actually analytically validate them on a different platform to make sure we're getting the right genotype, which is also important. We did that in the original trial, and that was 100% concordance, but we want to do it here as well. But in order to do this sort of analysis and do this sort of genotyping in the clinic, we need to store the kit at minus 20 degrees. So the freezers for minus 20 degrees were not available in clinics. So we had to buy freezers for those clinics and so on. But finally, in the Royal Liverpool Hospital, we secured some freezer space in a GCLP lab, and we needed to get an agreement letter so that we could actually do this and so on. Now, we also need, obviously, training on how you collect the samples, etc., and develop specific information. The samples are stored and then transferred for quality assurance purposes, as I said, and we've had letters written, etc., so that we don't need to have a material transfer agreement. And again, we have a standard operating procedure written for storage and transfer of samples, so that everything's done under standardized processes. Number 5. We have to have a data collection form. Obviously, to do data collection form, and if you're putting into in an implementation setting, you have to get the sort of appropriate people within the clinic to collect data because, it's implementations. We can't have research nurses, etc., being doing this. So we have to provide funding for the clinics to get the data. In order for them to upload the data, we have to upgrade servers, etc., and so on, and then put it into the database that we actually developed for them. We have to train staff on the use of the database. So again, that's another lot of training. We need to have a control group to compare to what we're doing in terms of the genetics. And so the ideal would be that we use different clinics in the same region, and we actually dump all the data, because all the data is a computer, but we can't do that. It's not allowed. It goes against data protection, we're told. So we actually have to do it on individual cases, anonymized and so on, that we can actually get a control set that we can compare to in terms of what we're doing. Number 6, 5 patients have been got onto the program. We have started, so that's at least we got there. Because the genotyping has to be done by a nurse and patient has to be given information, this has slowed the clinic down, and that is an issue. Because if you start introducing procedures, the kind of usual pathways will slow, and you need to think about how you can operationalize the pathways to improve the flow through clinic and so on, and we're working with them to see how we can do that. And clearly there are lots of patients waiting, and we don't want to increase the waiting times for patients as well. So we need to consider all of that, and that's a real concern if you are going to introduce a disruptive innovation into clinic, and we need to think about that. The problem that we're going to find is that anticoagulation patterns in clinical settings differ in different settings, and so all of the above I've told you about will need to be modified in different settings. Our aim is to collect 300 patients compared to equivalent number of patients in other sites not being implemented, and to see whether that actually improves things. This will include another cost-effective analysis. We've done it in a randomized control trial. Now we're going to do it in a real-world setting. If this all shows it can be implemented, I have no idea what the commissioners will say, but hopefully we can work with NHS England, etc., whether it needs to be rolled out or whether it can be rolled out. And obviously we are working with the HSN and clerks on this project to be able to do this. Now you may think, well, is this really worth it? Why don't we just change everybody to direct oral anticoagulants? Well, I've told you the reason why we can't, because of the cost issues per se. And in fact, a paper which has just come out, published online on April 20th, from a completely independent group, says that well-managed warfarin therapies associated with a low risk of complications as still a valid alternative for prophylaxis of atrial fibrillation associated stroke. Therapy should be closely monitored, etc., which is exactly what I'm telling you about. So this is where we're at, and that's why we think the warfarin is still a very important drug. We need to improve the way we use it. We need to think about how we can implement genetics into the clinics, but also give patients the choice in terms of when they need DOACs as well, rather than just say we don't use DOACs. So we need to be able to stratify therapy for the future. Thanks very much for your attention.